All right, so this will be hopefully a short video to try and simplify this one topic here. Let me grab black. So we are not going to worry about affinity. We're just going to get rid of that word. And what we are going to talk about is the delivery of oxygen. So down here we'll just scratch out all of this affinity stuff and not worry about it. So conceptually, one of the things you need to remember is that oxygen is carried by your red blood cells bound to hemoglobin. Right over here we see a little line of red blood cells, so you can assume that this is a little capillary or a blood vessel, I'm not drawing it very well, that runs through the heart tissue. So this red blood cell here is not going to cross that barrier, and these red blood cells here are not going to cross this barrier, and these here are going to stay in that tube wherever it goes. So if oxygen in a red blood cell is going to end up in a tissue, right, in one of these cells over here that needs it, then the hemoglobin is going to have to release the oxygen so that it can leave the bloodstream and enter the tissues. Remember, this is gas exchange. So hemoglobin stays in the red blood cell stays in the blood vessel. So oxygen, if it's going to get to the tissues, has to be released by the hemoglobin. Hemoglobin has to let go of the oxygen, and then it will diffuse into the tissues. So again here, the O2 has to leave the blood vessel, be released by the oxygen so that it can then enter the tissues. So the hemoglobin is holding on to the oxygen. There is binding. That's this affinity part, which we said we're not going to talk about. So if you want oxygen to get into the tissue out of the blood vessel, then it needs to be released by the oxygen, or excuse me, released by the hemoglobin, or delivered by the hemoglobin. So this relationship between hemoglobin and oxygen can be affected by these three factors here. The partial pressure due to oxygen in the tissue we are talking about, the temperature in the tissue we're talking about, and the pH in the tissue. So if we go back here, we're talking about the temperature, the pH, or the partial pressure due to oxygen in these cardiac cells. And that might be different than another part of the body. So what we're looking at here is which parts of the body are going to get more oxygen from the bloodstream than other parts of the body based on their physiology or their metabolic activity. So that your body is not just dropping off oxygen everywhere all the time and then having to reload all of its hemoglobin with oxygen all of the time. So you're sort of conserving the oxygen that you have bound to your hemoglobin. So the way this relationship between hemoglobin, we're getting rid of affinity, hemoglobin and oxygen works is that it works in such a way to encourage delivery of O2 in highly metabolic tissues. So then we need to look at partial pressure due to oxygen, temperature, and pH, and figure out what they are going to be in a highly metabolic tissue. So let's take, for instance, you are riding your bike up a hill. Your quadriceps are working very hard. Your legs are working very hard to power the bike. Your arms, not so much. So then, in the highly metabolic quadriceps, we want to think, are they going to have high partial pressure due to oxygen compared to like the biceps, or low partial pressure due to oxygen? And you want to think about not how much oxygen you want them to have, but how much oxygen they're going to have, given that they are highly metabolic and therefore using lots of oxygen. So, should your quadriceps have high partial pressure due to oxygen or low? If you say low, that is the correct answer. So, in highly metabolic tissues, there is low partial pressure due to oxygen. This increases the delivery of oxygen. In a highly metabolic tissue, would you expect its temperature to be high or low compared to a not highly metabolic tissue? So if you were able to stick a temperature probe in your quadriceps, would the temperature there be higher or lower than your biceps when you're riding a bike up a hill? If you're saying higher in the highly metabolic tissue, you are correct. This increases delivery. And then now think, same instance, you're riding your bike up a hill. You are pedaling really hard. The hill is steep and you are trying to get it fast. So your muscles have gone anaerobic and you are feeling the burn. Should the pH in your quadriceps be high or low compared to that in your biceps, which are not working nearly as hard? Right? You should think low, right? So the example I gave you with anaerobic muscles, they would be releasing lactic acid or producing lactic acid, which would make that tissue more acidic than a tissue that hasn't gone anaerobic. But even if you're just highly metabolic, there are metabolic byproducts other than lactic acid that tend to lower the pH in highly metabolic tissues. So low pH is going to increase delivery as well. And I'm just going to do like a few minutes, hopefully as quickly as possible, to talk about how this works or why it is this way. So you want to remember, if you've got one hemoglobin, it's 4 hemes. So these are not 4 red blood cells. These are the 4 hemes of a hemoglobin. So we're looking at one individual molecule of hemoglobin. The hemoglobin is a protein, and it should not be too difficult for us to conceptualize that changes in temperature might affect how a protein works, or changes in pH might affect how a protein works. Right? If you raise the temperature too high, you can denature a protein. You change its shape. pH does the same thing. If you've ever had ceviche, that is seafood, usually small chunks of light seafood like shrimp and scallops and whitefish, which they just let sit in lemon juice. And the citric acid from the lemon juice gently cooks the seafood, so you get this delicious ceviche. You might, if you've ever marinated chicken too long, like maybe you're going to make lemon garlic chicken or something, and you throw some lemon juice or some sort of acid in your marinade, and then you leave it in the refrigerator overnight, and the edges of the chicken turn white, you have cooked the chicken with the acid. We'll go over how that works later. So hopefully it's not too difficult to envision how temperature or pH, or changes in temperature and pH, could cause hemoglobin to let go of oxygen. So hopefully that's not too difficult. Understanding the partial pressure due to oxygen part is a little bit trickier. And what you need to understand is that each one of, or I should say all of the hemes affect one another. So how tightly heme number 1 holds on to this oxygen, or I should say, let me back up. The oxygenation status of heme number 1 affects how tightly heme number 2 is holding on to its oxygen. So the way it works is that if you have one of the hemes that has oxygen bound to it, that makes the other hemes hold on to their oxygen stronger. And then when you start losing oxygens, each of the oxygens on the remaining hemes are held a little bit looser. So if you're in a highly metabolic tissue, and I knew it was going to do that. Let me see, is there an eraser? Where's the eraser? If you're in a highly metabolic tissue and that heme lets go of its oxygen, there might be more oxygen around that would just hop back on. So it would stay more oxygenated. But if you are in a low metabolic, or excuse me, a high, what did I say? If you're in a, these are so hard to do without a script. If you are in a tissue with low metabolic rate, meaning it would then have lots of oxygen, when one oxygen leaves, another might take its place, is what I'm trying to say. Then imagine you are in a highly metabolic tissue where there isn't a lot of oxygen. So this oxygen gets released. It is going to diffuse away very quickly because there's no other oxygens for it to bump into, which would send it back towards the hemoglobin. So once this oxygen leaves, these three oxygens are now not held as tightly. The hemoglobin's grip on them has loosened. So it is more likely that this oxygen is going to leave and go away. And when this heme here is empty, these two are now holding on to the oxygen more loosely. So it's now easier for these two oxygens to escape. So every time an oxygen escapes, it makes it more likely that the next oxygen is going to escape. And it's more likely for an oxygen to escape and diffuse away under low partial pressure due to oxygen. So when there's not a lot of oxygen around, hemoglobin starts losing oxygens, and then once it starts losing oxygens, it just loses the next one, and the next one, and the next one, because the hemoglobin keeps loosening its grip on the oxygen every time it loses one of them. So what this does is encourage hemoglobin to let go of as much oxygen as possible when it finds itself in a tissue with low partial pressure due to oxygen. So it's kind of neat. Conversely, and this you don't really need to know, but now I want to say it, when this hemoglobin finds itself in the lungs, right? Whoop, sorry. Let me fix that. There we go. In the lungs, there's lots of oxygen. So once the first oxygen hops on, this hemoglobin is now even more attractive to other oxygens. It's like more magnetically oxygenic, if that even makes sense. So once one oxygen hops on, it makes the other hemes more attractive to oxygen. And then when this one binds, these two are now more attractive to oxygen, which makes it more likely that a third will bind. And when the third one binds, this heme becomes even more attractive to the oxygen. So this relationship between the four hemes, where the more oxygen you have, the more oxygen you want, and vice versa, right? As you lose oxygen, you're more likely to lose more, encourages hemoglobin to lose all of its oxygen in highly metabolic tissues, but also soak up a lot of oxygen real quick once it gets to the lungs. So it's pretty cool. So long story short, you need to know that more oxygen is going to be delivered in tissues with low PO2, low partial pressure due to oxygen, higher temperature, and lower pH. because those are the conditions in highly metabolic tissues, and you